Okay, well, thanks very much, Gamze, for the introduction, and thank you to the organizers for inviting me. I've really enjoyed this meeting. I think there's been all sorts of really high quality and thought-provoking presentations, and it's exciting to see so many young people and so many new ideas advancing the field, and it's necessary, as I'm going to tell you, because we absolutely need all this innovation computationally. Okay, so I'm, let's see. So I think we're still, I was involved in the original Human Genome Project. I'm old enough for that to have happened. Some of you may not have been born. When we finished that, people thought, okay, that's done. You know, we're going to move on to post-genomics. Actually, there was a term post-genomics for a period of time, and now we're post-genomics. I think we all, recognize the genomic era is ongoing. We also recognize that genome sequencing has been a transformative technology for biology. But I would argue there's still a sort of sense that we're going on to think about function and other things, and that the genome sequencing part of it is finished, and I don't think that's the case. We're still in the middle of it, as far as I'm concerned, and it's still developing and has new applications. And it's really about that I'm going to be talking today, and try and enthuse you about the current and future era of genome sequencing. And I think of it as going in three phases. The first one was the reference genomes for key organisms. That gave us the human, sort of culminated in the human genome, or a few years after there was the mouse and rice and Arabidopsis and a few organisms. And that was done at great cost, with big efforts and so on, using Sanger technology, which had been invented and was basically the technology that enabled DNA sequencing to start with in the late 1970s, that was automated in machines. And then we had a phase two, which was dominated technologically by short reads, and that enabled resequencing. So it enabled mapping to reference genomes, and that was both for population variation, and I was very involved in that, as Gamze said, in human genetic variation. But also enabled all the star-seq, the ChIP-seq, REF-seq, ATAC-seq, RNA-seq, of course, and so on. And that built on the references. And people did do some more reference genomes with short reads, but that was always unsatisfactory. Essentially, they're too short to assemble well. So, Ewan talked about this yesterday, or Monday. Now we're in a third phase. We can ab initio sequence arbitrary genomes, and we can also sequence arbitrary nucleic acids in their native form using the single molecule technologies. And I think we're still at the start of that phase. It's actually, people have had these instruments for some time, but they're really coming into their own now, or in the last year or two, as workhorses. There's this concept that they're much more costly than Illumina, but that's actually not true. The per base raw cost is getting down to being quite similar to Illumina. It's the same order of magnitude, especially at scale. And the trajectory is much better. And so within a year or two, we're going to have cheaper data from long-read technologies. And in a way, it's not surprising. I mean, the reagent use is obviously less. You're going down to the core minimal object. So I think this is going to dominate the next decade. And this advance has been pretty staggering. We're kind of used to these exponentially increasing technologies. Actually, the first time I sort of heard this analysis was from Sidney Brenner, who was a very visionary and witty guy. Together, and he did a sort of speaking tour with Nathan Myhrvold, who'd been the chief technology officer for Microsoft in 2005. And they pointed out that DNA sequencing is an information technology. It's not really a biological technology. And let's see. Yeah, so, oh, God. Yeah, so there's been a 10 to the 9 increase in scale from a million to a million genomes, which we've basically got to a couple of years ago, and at least a 10 to the 7 drop in price. 10 to the 9 is about 2 to the 30. And that's less than 30 years. So it's more than twofold a year increase in data, and that's substantially faster than Moore's law. And so, actually, as I mentioned in the discussion on Monday afternoon, when we sequenced the human genome, we were using quite clunky software and informatics, and we had to build very, very big compute farms. We were at the bleeding edge of compute resource in science. We had comparable data systems to CERN, for example, as part of the Human Genome Project. That's no longer true, and the reason for that is because of all your work and the algorithmic work. There's been a continual demand for computational improvement. And I see this as a sort of ongoing process of interplay between the analysis and understanding of gene function. That's one thread in this talk. Improved computational and mathematical methods. That's another thread. But also evolutionary models. We've heard something about that, but actually I think it's absolutely central to our understanding of DNA and where DNA comes from and biology. So in any case, DNA, sequence, genomes, and computation all go together. We're at the heart of this biological revolution in this room. And a sort of idea to throw out there is, when I was a graduate student, I used to be a bit envious. I came from a maths background originally. I used to be envious of physics. Physicists have mathematics as a kind of theory, and theoretical physics, and it's very beautiful. And I've come to realize that informatics, actually I didn't like the word bioinformatics originally. I like the kind of concept of theoretical biology or computational biology. But I've come to realize that information is in some sense to biology what mathematics is to physics. It's very fundamental. DNA is information that's transmitted, and all this characterization of flow of information in cells, it's both scientifically central and analytically critical. So maybe we should be embracing computer science as the theory behind biology. Okay, so this slide is a bit redundant. We've heard from Ewan already about single molecule sequencing advantages. In a key position, he was saying that ONT can now get to more than a megabase. But really the critical thing to say is that it's arbitrary. You can, in principle, sequence as long a molecule as you can put into the system. So more than 10 kilobases is absolutely standard. All these technologies do that. It's 2 orders of magnitude longer than standard short-read sequencing. 100 kb, bigger than 100 kb is common. We think of that for Oxford Nanopore, but actually PacBio reads, they run round the molecule, say 10 times. If you're getting a 15 kb PacBio hi-fi read, it's 150 bases, 150 kilobases, the actual read. You're just using it to sequence the same thing 10 times or 8 times or whatever. And so both technologies do this, and over a megabase is possible, but it's demanding on the input. So we're really limited by input rather than technology. And the reason for that actually is in the next line. In the first and second phases, you had to sequence collections of molecules, and it was very hard to keep them coherent. And you lost coherence as you went along the sequence, and that limited the read length. But of course, with a single molecule, there isn't that problem. The cost is dropping. I've talked about this. PacBio are just introducing this year a scaling which allows under $1,000 for a 30x human genome. So they're chasing that, even in their hi-fi. In their raw data, it's cheaper than Illumina, though in some sense you don't see that. And we can directly assay the source material. Ewan talked about methylation and arbitrary DNA and RNA modifications, but the error rates have been decreasing, and now the ONT duplex is well under 1% as well. So the future is bright, and I think that is going to continue to improve. Those technologies are still on a exponentially improving trajectory. So what are the consequences for assembly, which is, well, this is an old slide. This is at some point in 2018 or 17, at the end of 2017, I took all the genome assemblies in GenBank, and I plotted their initial submission date on the x-axis and the contigen 50, which is the typical size of the piece that you assemble, on the y-axis, both, and the y-axis is in log space. And what you'll see is that they kind of come in 3 periods. There was an initial period that was the Sanger sequence where we got human, mouse, and chicken, and then a few others. It was extremely expensive, as I said, and quite limited, and there's a scattering of sequences. And then there was a period with a lot of assemblies, but they really weren't very good. They were short, and the typical size here is in the 10 kb range, and that's the same size as genes, and so lots and lots of genes were fragmented, and there were also problems of repeats and repetitive genes, not just transposons and potential junk, but repetitive functional material and disentangling that. And then you can see that suddenly around here we started to get a new class of assembly, and that was due to long reads. And they were introduced in 2015 or 16, but that has just been accelerating. And a good illustration for me, and something that should have changed my mind about this and changed the direction of what I was doing scientifically, was that I had worked hard to assemble the gorilla genome. That seemed like a good goal after the human in 2000, and it took a long time, a decade. And we started off with Sanger, we switched to Illumina, we used a mixture of different technologies, and we ended up with a 12 kilobase contigen 50. And a lot of people were involved. It was one of these international consortia. And this is the gorilla that we sequenced from San Diego Zoo. And in 2016, as a kind of demonstrator, PacBio, the company, collaborated with Evan Eichler's group, and in a year, basically, in a small, tight effort, they got 75x coverage with their old technology, and they got an assembly that was nearly 1,000 times longer in continuity, and that made a substantial difference. So this is a picture from their, can you see my mouse? No. Let me see. Okay, well, this is a picture from their paper. The black bars underneath the chromosomes indicate the sizes of the contiguously assembled pieces. Not only did they get much more contiguity, but they added 200 megabases of DNA, which was getting on over 5%, between 5 and 10%. And they made a whole lot of corrections. And we knew previously, by comparison to human, that about 15 to 20% of our genes were incomplete or duplicated or incorrect in some way. And so that's a substantial burden. That was the typical of a short read assembled genome. That dropped to 220 genes, which were incomplete in that assembly, which is about 1%, which is a tolerable level. I mean, that's a really substantial and significant difference. Okay, so based on that, some people started to say, we're now ready to go and apply this across the board. And what does that mean? What does it mean to scale it up? Well, genomics is always ambitious, and it should be ambitious. If we're living in an exponential space, we can think about not just increasing by a factor of 2, but by increasing by a factor of 1,000. That is typical for what can be achieved, at least on a decade time frame, and a decade isn't all that long in science. So people started to look at how many species are there. Well, there were catalogs of eukaryotic species, and those catalogs, it was somewhere between 1.5 and 2 million species. Now, everyone says that's an underestimate. I'm sure it is in some sense. There are all sorts of issues. What is a species? Is something seen once in 1875? Is that still a species? But people keep finding new ones each year. But there's of that order of species. And it's conceivable to think of sequencing everything, of getting a reference for everything. And that's because at 2-fold increase per year, We've done about 1,000 species. We'll come to that now. 2 to the 10 is 1,000. 1,000 times 1,000 is 1,000,000. We can conceive of sequencing 1,000,000 species. And if we can conceive of it, we should do it. It's almost, there's an imperative there. I mean, there's a phenomenal difference in getting completeness. That was a key message from each of the stages of the Genome Project. The human genome, in getting completeness, we could make complete lists of genes. We just, all of you in the room, assume that you have one of everything. You have all the genes, you can put them on a chip, or you can assay them, and you can map to the whole genome. You don't want to map to 60% of the genome. We want to map to the whole genome. And similarly, if we want to interact with the living world, not just humans, we're interested not just in humans, we're interested in our environment, and also not just model organisms. We can do that using genomics if we invest in genomic references for everything. So my belief is that this will connect modern molecular genetics to our interactions with the whole living world. It'll provide new models for gene function. It'll connect ecology to molecular genetics. It'll impact conservation, of course, studies of evolution, but also bioengineering. It'll provide vast numbers of examples of different uses and applications of biological pathways, and a whole lot of other things. So really, this is a major endeavor. I think it's at least as important as sequencing the human reference, and we're just starting. So that's in some sense. Okay, so what did that entail? Well, the Earth Biogenome Project is really a vision, and what's happened is that there's been a whole set of projects start within its space, which are kind of coordinated through it, and the coordination is quite good. And I'm going to talk in particular about this Darwin Tree of Life project, which was started within the UK by the Sanger Institute. It's now part of a larger effort, ERGA, the European Reference Genome Atlas, which is an EU-funded consortium to coordinate reference genome sequencing of all of life across Europe. But DTOL started in 2019, and the aim was, in some sense, how do you partition this task? And a natural way to do that is to do it by geographic location. You might think you want to do it phylogenetically, but then what happens is everybody wants to do the phylogenetically most important species. And you get people globally kind of stepping over each other, trying to race to sequence the same things. So there are other reasons. I know it sounds a bit Brexity. I'm always slightly nervous, but we're very much not Brexity. Everything is totally open. We're collaborating with all the other efforts around the world. It's easy to coordinate. There's a huge wealth of knowledge and a set of natural partners in Britain. And this isn't just a genomics project. We have to collect samples, just as medical genetics involved in genomics involved medicine. Diversity genomics involves working with the diversity partners. The Natural History Museum, Kew Gardens, these are world-leading institutions who've in detail cataloged and have samples of all the plant species in Britain, for example. It simplifies the logistics. It postpones ethical and legal questions about moving samples between countries and the natural rights of countries to their biodiversity. But we're very much embedded into broader efforts. Also, although Britain, 70,000, there are 70,000 identified species in Britain. It's probably amongst the best catalogued biotas on the planet. That's only 4% of the total number of catalogued species here. But if we go up to higher levels, it's nearly 40% of all the families are represented. So there are 10,000 families, and about 40% of them are found in Britain. It's 70% of all the orders, and 90% of all the phyla. So, at the levels which we can cover quite rapidly, we can have a substantial impact and link into other people's efforts elsewhere in the world. And of course, also, the British biota is shared almost completely. We're a sort of post-glacial appendage to Europe that happened to get separated by sea as the oceans rose. It's shared with the rest of Europe, and we are absolutely coordinating with other people in Europe, so we won't end up sequencing it all. It's going to be done collectively with efforts across the rest of Europe, and indeed beyond. Okay, so I'm going to tell you a bit about where we've got to in this project so far, and then some of the details about what it's entailed computationally. So as of last night, we had 660 submitted. That's about 1%. So we'd gone from 0.1% to 1% of the total, which is good. It's a factor of... but as you can see here, we've got complete data for another half as many again. So that would take us over 1,000. And then we've got some data for 2,000, and I think we've got 15,000 species now collected with samples in our database. And they come from across the tree of life. So here we have protists. Oops, let's see. Protists. These are plants. These are mosses and liverworts, flowering plants, fungi. These are jellyfish. These are the animals. Chordates form a relatively rich group here. Mollusks and annelids. These are crustacea, and then we're getting into the arthropods. The rest of them are arthropods. There are a lot of arthropods. Somebody said that God loves beetles. So there's quite a lot of beetles. Those are Coleoptera, but actually I think the most are Lepidoptera. So a lot of insects here. Okay, so, and in some ways these are easy to collect. They're kind of nice sized for sequencing. And also they're very good ecological and climate change and markers. And so immediately we're connecting to those communities. So in order to do this, we've put together a whole kind of what Mark Blackster, who's the PI for the project, calls the Tree of Life Genome Engine. And there's a lot of pieces to this, and actually there's a lot of scope for computation at multiple points in this, which we're going to get into. So we start by acquiring samples. We have to track that. We get a barcode. Although there's only of the order of 1,000 or a low number of thousands of good high-quality genome sequences, there's about 500,000 species have got barcodes, which is a short, so 600 base sequence from the mitochondrial CO1 gene. And so we can use that both to validate and also to track and connect to those efforts. We extract DNA, we sequence, we submit the raw data. We then assemble somewhere in here. Oh yeah, quality assurance, primary assembly, scaffolding, curation. I'm going to talk more about these steps. We submit all the data, the primary data and the assembly. It's annotated with Ensemble. We also do RNA sequencing. There's an open data portal. We're automating the generation of scientific papers so as to have a tiny little genome note, not in a high-profile journal, but in a sort of, but that's something that's citable and referenceable and contains, so that we can give credit to the people who collected the samples, the appropriate people, and that people who use it can feel uninhibited about using the data. So the aim is to do that rapidly, and then make a fully open resource that can be integrated into other people's and used as people want to as a platform for further research. Okay, so let's dig into assembly as the sort of first critical task, and one that I've been involved in overseeing. So our goal is chromosomal haplotype resolved assembly. And most eukaryotes are diploid, some are tetraploid or hexaploid. And you've got two copies of the genome, and up until recently we used to think that, okay, that we're just looking for one genome. But actually, there really are, if you're going to do a proper assembly, there are two copies. So the human genome is 6 gigabases, not 3 in any human. We want to resolve that because it turns out that's important, as we'll get back to. We want high contiguity, the standard that's in the field that's now established, and there's sort of consensus standards in the EBP is over a megabase, N50, and over Q40, QV. Actually, we're getting now into the Q50s and Q60s. That means less than 10 to the minus 4 error rate. We're approaching 1 in a million base error rate for many assemblies, or I think probably the mode is around, or the median is around 10 to the minus 5. So how to get there has been changing rapidly. It's only 3 or 4 years ago, in 2019, there's a figure at the left-hand side. There's a pipeline. You don't have to read it all, but these are all different data types. There are 4 different data types. We had to collect quite a lot of data. You can see these 60x, 50, 200, 50x. That's the coverage in raw data. So it's quite complex to collect that data. And then this is a kind of flow chart of the analysis. Everything in red is a program, so there's half a dozen or more different pieces of software involved, and including separating out things into separate haplotypes, and then scaffolding and recombining and integrating information again, and so on. So that was used to produce, there was a landmark paper in Nature a couple of years ago with, I think, 13 genomes from the Vertebrate Genomes Project, who really pushed this pipeline that was aiming to scale these goals. But by last year, we'd replaced every single piece of this diagram. So we're now doing just 2 types of data, Pacific Biosciences hi-fi data, at about 15x per haplotype. I think you should think, people talk about 30x for the human genome, but actually you should think of it per haplotype, per copy. That's the right way, because as you have different numbers of copies of the genome, you want more data. It's not true that you want 30x. If you have a tetraploid, you need 60x data. We have a new assembler, which Huang and Hung Li produced, hi-fi ASM, that we've been using. We have a new program, which I'm going to mention briefly, which we need as a kind of QC and correction step after hi-fi ASM still, but that, I think, is in the process of changing. So this was an innovation about 2 or 3 years ago, which maybe its life has come to an end already. And then following the initial assembly, we get a lot of pieces, contigs, but they aren't chromosomal. They aren't in chromosomes. We want to put them into chromosomes, and we're doing that with high-C data, and I'm going to talk about this a bit. We have a new scaffolder for this that I'm going to talk about. And then we go through some curation, manual curation. But as we're scaling up, and we're now submitting over an assembly a day, we can't have people go by hand through everything. So there's a philosophy, though, that we have that rather than say, oh, everything has to be automated, our goal is to carry on reducing the amount of human intervention so that we can go from needing a week per genome to a day per genome to an hour per genome, and then maybe at some point to 10 minutes per genome. But it's always worth looking, because when you look, you can see what is odd, you can pick things up, what needs to be fixed, and you can give feedback to the systems. Okay, so that's kind of where we are now. There is a future, which is possibly this T2T, complete, perfect, end-to-end, accurate assembly, maybe not even needing these steps. And I'm going to mention that briefly. I'm going to run out of time. So occasionally, even with our current approaches, we get complete contigs. Here's an example from a butterfly. We get a contig from end to end, and it's got telomeres at both ends, and it's correct, I can tell you. This is evidence about the telomeres. I'm not going to go into that. So we do assemble complete contigs through centromeres. In fact, we assemble centromeres quite often. Why aren't assemblies perfect? Almost every location is covered 20 times. They're not perfect because of repeats. We know repeats create trouble for assemblies. Paralogous repeats have different locations, and orthologous repeats. But the biggest problem is orthologous repeats, is heterozygosity. Really, I told you we've got 2 version copies of the genome. That's a big repeat. It's a repeat on the scale of 100 megabases. And that makes life particularly hard, and that's why people didn't want to think about it in the past. But it creates a problem. In vertebrates, in humans, it's about 0.1% heterozygosity. We're not so different, but vertebrates go up to 1%, many insects up to 5%, and plants. And at that level of diversity, all these structural variants that we were hearing about are major issues. So there are regions of high similarity and regions of high variation, and it can get up to 15%. So we need to keep things separate, and in fact, the only good historical genomes were haploid or clonal. Fly, yeast, C. elegans were all clonal, and human was done from cloned fragments. And even in good new assemblies, most errors and contig breaks are caused by heterozygosity. So this was one from the Vertebrate Genomes Project. Really, I just want to show there's a piece here that's duplicated. And it duplicates a gene. It's not just a... And what's going on there? Well, what's going on is that there are two pieces of assembly, one coming from the left and one coming from the right. And when they meet, and the assembler tries to kind of put them together, it overshoots because they're somewhat divergent. And so you end up with the piece that overlaps being there twice in the final version. Okay. So how do we deal with that? Well, one strategy is to use genetics, to use trios, to trace things coming from the maternal and paternal chromosome. Sergei Koren and Adam Philippi did this spectacularly with a cross between two different sorts of cow. We'd also proposed this idea earlier, but not based on long-read data. And suddenly they got an improvement of about tenfold in contiguity in doing this. And we did the same down here. You can't see with zebrafish, where we made a cross between strains. So when you can set up a cross and you can follow genetic variants through ancestors, you can combine genetic information, then you can use that to tease apart the two haplotypes. But you can't do that in most cases because we get a sample from the wild and we don't know the parents. So what can we do in that case? Well, actually, there are telltale signs of these problems, of these overlap duplications. And here's an example from Anopheles, from mosquitoes. And here we've got, in black, one contigs from one, so we've got the truth on the x-axis, and we've got the assembly on the y-axis. The assembly came in 3 contigs, and you can see that there's an overlap in the contigs at both ends. And in those overlaps, if we look at the read mapping, then there's only half the read depth, because the reads in these regions, the reads from both haplotypes map to the same contig, but in the overlap regions, they map to their appropriate contig. So you suddenly drop to half depth, and also the overlap is similar to itself. So we can use those sorts of signals and a variety of heuristics to remove the duplications, and there have been a number of different strategies, and Dongfang Guan, when he was a visiting student with me, developed this program Purge Dupes, which is now used essentially by all the large high-throughput assembly projects. And it's a bit of a kludge, but it does it deals with 90% of the problem. And we reduce the number of duplicates by 5-fold in this case, and remove about 15% of the data, which in this case happens to come from duplicates. And you can also see that in this plot, if you understand these plots, these two peaks should overlap exactly, which they do after analysis, but they don't beforehand. So there was excess duplication here. Okay, so that's one issue, the scope for improvement there. Another way to do that is actually to use additional data in the graph and to use a more graph-based analysis. And Shilpa Garg, who I think has left but was at this meeting, started, I think, working with Tobias Marschall, and also was a visitor to me for a time, approach to this. The latest assemblers do this. Sahayu Chang, Hi-Fiasm, can integrate Hi-C into the assembly graph and thread. And this is, in some sense, how the T2T genome was put together, with ultra-long ONT reads from Adam Philippi's group. And that haplotype resolved all by threading through the graph and involving Mikko Raitinen's methods to pull apart the haplotypes. We also heard about this, I think, yesterday. So ultimately, this is the right way to do it, but it's still, I would say, not really production mainstream, although The Philippi group is pushing this into other species. But doing it their way is quite expensive and heavy. It's not scalable to the EBP scale. Okay, so that's dealing with duplication. What about scaffolding? I said we want chromosomes. Well, this is an example, nice example. This is a cricket. It's got a 9-gig genome, 13 chromosomes. These are, it's got quite a nice contigand 50 of 10 megabases, but you know, the chromosomes are going to be hundreds of megabases. So they're still fragments of chromosomes. And this is a high C plot, so we have an X and a Y axis, and you get density, like red density, wherever there are high C links between reads. So along the diagonal, you can see the contigs, but there's a lot of small contigs here, and there's a lot of possible connections that can be made. So this is actually not the raw contigs. This is after using SALSA 2, which was the state-of-the-art scaffolder. So the goal is to put this together into a set of chromosomes. This was an advance in 2021, PINS, which you can see is kind of dealt with some of this small pieces, but still has lots of stuff off diagonal, which reflects incompletely sorted out links. And Chen-Shi Zhou, who's a postdoc with me, has introduced this new scaffolder that's published earlier this year, YARS, which ends up with this. So these basically are the chromosomes. There are 13 chromosomes here. This one that's pale is the X chromosome. It's at half coverage. This is the, and what he's doing is looking within an assembled, and there's very little off the diagonal. There's a little piece here, so this piece probably belongs attached at the end here. So there's a few things which are incomplete, but it's essentially complete. It's looking at, in regions which are trustworthy and consistent, what is the high C pattern off the diagonal. And it builds a model of that, and then it tries to look to move pieces around in the order and glue them together so as to satisfy this model across other regions. So there's a statistical component to this. Okay, so this is pretty good, not complete. There's actually still some errors. There's an error which probably should be broken, and there's some sort of join here. So there's something going on here. There's some sort of error here. That's the X chromosome. This is another example that looks nice. And we've dealt with most things. There's also still some pieces down here, and there's still some signal in the small pieces which hasn't been dealt with. This is our biggest genome so far. We thought we'd get a 100 gigabase genome. There are a few species with 100 gigabase genomes. There are plants with 150 gigabase genomes. Mistletoe, it turned out to be 99.5 gigs, very frustrating, but this is the biggest genome assembled so far. It's assembled down to chromosomes. It's kind of amazing to me that with modern hi-fi data, you can put it in, and hi-fi-asm just delivered this. You had to wait a bit longer, but it delivered it. Other tools had to be re-engineered to handle it, the scale. You get this nice crisscross pattern. That's a biological phenomenon. That's because of the way the chromosomes are arranged in the cell. They're so big, they have to be packed in interesting ways. So this is what YARS delivered. It's not bad, but there's quite a lot of error in there. And this was done with some manual curation, which actually took quite a long time in this case. So there's actually an open problem for you all about how to improve this further. And we think that really machine learning it's very sort of vision type problem. It should be that we can learn how to scaffold genomes. And that's an open problem. As we scale, we want to spend less time fixing errors. Okay. Here's another issue that's come up. So plant organelles. So animals have a mitochondrion as well as a autosomal genome. And people ignore the mitochondrion often. It's very simple. It's 16 kb. In fact, single hi-fi reads run around it. It's very easy to assemble. Still, we had a sort of specific pipeline that's here is in bioRxiv called MitoHiFi. They're led by Marcella at the Sanger Institute and partners across the world. But they're very standard. You can know whether or not you've got it right. There are problems because there are pieces of mitochondrial DNA in the nucleus, and you have to separate from those, and so that In the assembly graph, you have to separate out the true thing. But in general, you can assemble animal mitochondria. But we were sort of naively, didn't know much about, I mean, one thing one keeps learning about the biological world. Plants have two organelles, of course, we knew that, plastids, the where light is handled. Sorry, there's another word I'm blanking on. And mitochondria, of course. But these are much more complex genetically. So plastids are 1 to 200 kb, and that's the typical structure. They have a long inverted repeat. And it's 10 kb, so you might think you get a long read, you can span across that, but what you do is you find that reads go across in both directions. They go through here and around that way and like that, or they go through the top and around this way. And that's because there's recombination going on inside the cell. So actually the graph is a representation of what is present in the cell. In this case, it's not a case that there's a graph which you have to simplify. And then if we look at mitochondria, plant mitochondria vary between 200 and 200 kb and 2 megabases. And they come in all sorts of shapes and sizes. This is a simple one shown here. So here we have a non-simple graph. And again, there are quite short linker sequences here, but they're duplicated, and you find reads that follow all paths through this. So because of this, standard assemblers get confused. And frankly, if you apply HiFiism or HiCanoe or any off-the-shelf long read assembler, they get tangled up in plant organelle genomes. So Chen-Shi also developed a new package for this, OAT-K. It's got a new assembler that's based on syncmers that's actually somewhat related to Barris, Bonnie, and Ryan's MDBG. And he's been applying this systematically, and we can apply this early because we don't need high-C data, so we've assembled nearly 400 plastids and 170 mitochondria. And all sorts of interesting things come out. There's a variety in sizes. This is the distribution of genome sizes. A couple of small plastids. Those are in plants that have lost photosynthesis. They still keep a plastid, so it's still doing some cellular functions, but of course it doesn't need all the light harvesting stuff. Some plants appear to have multiple plastids, which are genetically distinct, complete copies. So there's some internal competition synergy that I knew nothing about before. You get fragmented genomes. You get, here's a tree, alder. It's got a circular component to its mitochondrion, but it's also got a linear component. This is linear, and this is the telomeric structure, which kind of has a duplication which creates a loop in the graph. So you get ones that are fragmented into pieces. That was already known, but they, of course, lead to assembly issues. And you also get cases where there have been pieces shared between mitochondria and plastids, and there appears to be recombination and much more complicated graph structures. And how to represent these properly is an issue, how to present them to the world, how people should compute with them. Should we linearize them or circularize them? You know, what does it mean? Copy number of the various pieces may vary. Also, though, once we start doing this, and there is this variation, because we're looking across hundreds of species, we can start digging into interesting biology. So we can, here we can do a sort of gene by species plot, and we can bicluster, and we find interesting patterns of duplication and loss. Yeah, the colors have kind of gone here, but there are sets of species with shared patterns of loss or gain of particular genes, and these imply bringing together association of function across species. And there's a number, I think there's going to be an increasing opportunity for looking at correlated evolution, essentially. So this is just one example, and I'm going to show another example as my final, or in a minute. So it's opening up new opportunities to look at this sort of structure. Over here, we have a set of things that are missing in most plants, but actually these are mosses and liverworts, which are very deeply divergent, which have a new set of genes, whole set of functions in in their plastid, not in other plants. So here's another example of the same sort of thing that Emmeline Vankester in Mark Blackster's group has looked at. As well as sequencing the host, we also get any symbiont that comes along with it. And so across all the insects and arthropods, many of them contain Wolbachia. This is an intracellular bacterial symbiont. And in some sense, people think of this as being inherited through the maternal line in the egg. And there's interesting biology associated with that. So people had previously studied Wolbachia diversity, and they'd got of the order of 100 Wolbachias. We've doubled that just as a byproduct of the detail project so far. And you get a complete survey. It's also, in some sense, unbiased. So here, Wolbachia comes in two supergroups, A and B. And here you see the relationship between the Wolbachia strains at the top, and this is the phylogeny of the species that which are the host species at the bottom. And you can see these are extremely different. There's been a lot of horizontal transfer. There have been some big expansions and bursts here of particular sets, but some of these have also jumped, obviously, very recently, all the way from Diptera to Lepidoptera, which is a big phylogenetic jump. So there's a lot of interesting potential biology coming out of that type of approach. Okay, I'm going to finish off by talking about plant centromeres. I said before we could sequence through centromeres. One of the things that actually drove the T2T effort was Karen Meager's interest in satellite DNA and centromeric sequence, which now we can completely sequence in humans. But we can do that across all these other species. And typically, you may think, I mean, at least I thought, that centromeres had long tandem copies of these arrays of repeated sequence. And here's a classic example. This is Arabidopsis. It's got 5 chromosomes. Each have got a big red block. And there's a pipeline called TRASH from Ian Henderson's group in Cambridge in plant sciences, which looks for these. And in this picture, you're seeing these big red clusters. And there's also some green clusters nearby of tandem repeats. There's 1 per chromosome, basically. This is the leftover scaffolds, and they've got some sort of other repeat, but there's only 2 megabases of that. Okay, so that's one case. Michael Hong, who was a master's student, applied this across all the 200 detail species at the time, all across the tree of life, and saw all sorts of different patterns. So, in some cases, we get a clean, this is oak, we get 1 nice cluster per chromosome, except for this chromosome, a small one, I guess. Here's an organism which is holocentric. You can't see, but there's little red lines all over this. So there was a paper in Cell, actually, about holocentric plants. They have dispersed satellite repeats. This is a plant with very different sequences on each chromosome. Some chromosomes have nothing on them, so there's something odd going on here. Some are near the telomeres, some near the centromeres. It looks like the whole standard centromere pattern is breaking down. There's a very interesting pattern here where there's something at the centromeres and the telomeres. But some species had no discernible satellite repeats. There was no tandem duplication, no sign of a centromeric repeat. So we wondered what was going on, and Michael started looking at transposable elements. And he came up with a cool idea, which is that for each transposable element family, he looked at where it was found in the genome. So you can think of a matrix of family on one axis and genomic location on the other axis. So family on one axis, genomic location on the other. And then you did a PCA plot of those matrices. Okay, coming to the end. And the PCA plot is shown here, and there's a dot per family. Most families are in this cluster. But there's this long tail here. So some families are very different, and this is PC1, have very different genomic distributions. And then there's a kind of, a tail of ones that are running back to this distribution. So what's going on? Well, what's going on is that these ones that are at the tail, at the tip, are in clusters, and they're in a single cluster per chromosome. So there's a few transposons which are forming these very tight clusters in the middle of each chromosome. And the ones that are along this tail halfway are sort of spreading out from that. And what's happened with a bit more investigation is that there's transposons inserting at a single point, which we believe to be the centromere, and new transposon families keep invading. There's an evolutionary arms race, and then they're pushing apart the earlier ones that invaded, and the earlier ones are kind of in this tail down here. And so what happens is the ones which are most at the edge are most conserved. They're the youngest ones. As you go backwards along the tail, they get less and less conserved. This is from 95 to 80% conserved. And if we look at what they are, they fall in one family, but they're sort of scattered across this family. They're all LTR transposable elements, but it's not like it's always a related element. It's a kind of new element from that class. So there's a lot of interesting, biology of the evolution of centromeres that we can pull out with this. And he can run that across all the species. And what we see is that there's been this transition between centromeric repeat types from satellite repeats, from tandem repeats to transposable element nests. And also there's been repeated switches to holocentric. That's completely distributed centromeric function. So centromeric evolution is incredibly rapid and dynamic, and we can now look at it with all these new genome sequences, both across species and within species. Okay, so I'll finish there. It's just a few key takeaways I want to get across. So first is that advances in sequencing technologies show no sign of slowing, and progress relies on corresponding advances in computational methods and tools. The second is that long-read sequencing allows efficient, accurate whole genome assembly, arguably for the first time. And the world of all macroscopic species is finite, and so we can, as far as I'm concerned, we will. It's going to happen. It's just as there's this natural imperative to complete the story. How we do it matters. To provide reference sequences for all large-scale life. I'm not so sure about viruses and microbial life. That's another world. This next phase of genome sequencing is underway. All aspects of it are changing very rapidly. There's a lot of scope of innovation. I really think there's a lot of opportunity for people to contribute in all sorts of ways. And sometimes that's in a fundamental method advance, and sometimes it's in tailoring, understanding what are the blocking points and fixing those. And that often involves innovation as well. It's often the detail provides reasons for innovation, not just the main problem of how do you do assembly. And then finally, that having thousands of references enables many new analyses. It's within and between genomes. And I just kind of scratched the surface. I mean, I've got a small group. We only look at certain things. There's going to be vast amounts of interesting comparative biology to be done, and I think the future is rich. So there's my group. On one side, it's in the sort of rainy Cambridge. There's Chen Shi and Michael. But we also have fun. This is us punting on the cam in Cambridge in the summer. And I'm, anybody who's interested in being part of this big adventure, I'm very open to people coming at a postdoc or student level. So lots of chances. Thanks.